HE double hockey stick has happened. Like, and my mom's screaming, "Oh my
Okay, I think everybody can hear me, and we're going to continue our discussion on muscles. We're going to, we're up to the point where we know what an action potential is. At least I hope we do. And I want to go over it again anyway. But how do we get to a contraction? That's the question I'm sure I left you with, and you've probably pondered that all weekend. So, okay. Does anybody have any questions about anything before I open this up? Okay, let's go here to, this is the last place we were on Thursday. Talking about an action potential. An action potential is another term for nerve impulse. Action potential is more precise than a nerve impulse because what we're doing, all cells have a charge on their membrane. We call that the resting membrane potential. Muscle cells are minus 90 millivolts. Nerve cells are minus 70 millivolts. All the other ones sort of fall in between. The action potential is what occurs when we change that charge. When we change the resting charge and we raise it or lower it. And that's the action potential. And so when we get an action potential, we send a wave of energy essentially across the surface of the cell membrane and into the cell to do something. Whether you're a nerve cell sending a signal down your axon, that's an action potential, or a muscle cell getting ready to contract. Now, a couple terms here are very important to us. Of course, the resting potential of a muscle cell is minus 90 millivolts. Commit that to memory. I seriously doubt if anybody will ever come up to you and say, hey, what's the resting membrane potential of a muscle cell? However, you never know. We're getting into it. We're starting to move into areas where some of these questions could possibly get asked as you go along in healthcare. So the resting membrane potential is minus 90 millivolts. Term that's important is threshold. In the case of the muscle cell, threshold is minus 50 millivolts. How do we get to threshold? Well, what will happen is on a muscle cell, we'll have an action potential come down a motor nerve that will cause the release of acetylcholine at an axon terminal. Acetylcholine drifts across the synaptic cleft at the neuromuscular junction. That's where the axon terminal is in contact with the muscle cell. Acetylcholine, the neurotransmitter, will land on the motor end plate on the muscle cell. There's only one location on a muscle cell called the motor end plate. This is where you have sodium gates that are controlled by acetylcholine. These are called ligand gates or chemical gates. The acetylcholine lands on receptor sites on these gates. These gates open up. Sodium goes into the muscle cell at the motor end plate. And we start changing the charge of the motor end plate because the whole muscle cell is minus 90 millivolts. Sodium is a positive ion. It has a little plus next to it. So it's a positive ion. Sodium goes across, goes into the motor end plate from the synaptic cleft. It's all in the interstitial fluid outside the muscle cell. We start changing the charge at the motor end plate. We have what we call a localized depolarization. The motor end plate goes from minus 90 to minus 80 to minus 70 to minus 60. And if we get to minus 50, which is threshold, that value, that means the charge on the motor end plate has changed from minus 90 to minus 50 millivolts. It has gotten more positive, and there are electrical gates on either side of the motor end plate for sodium and for potassium that will now get affected by the change in the charge. So we depolarize the motor end plate if we hit threshold, then voltage gates, electrical gates on either side for sodium will open up, and you'll have a massive rush of sodium into the muscle cell, depolarizing the muscle cell. Now this is an incredible inrush of sodium. We have a depolarization event. Once it starts, it doesn't stop. This is your action potential. Now, that sodium gate adjacent to the motor end plate will stay open until the charge changes from -50, that was threshold, all the way up to +30. We get to +30, the sodium gates shut and the potassium gates open. Potassium gates open at +30 and they stay open all the way back down to -90 millivolts. What we're doing now is repolarizing. We depolarize and then we repolarize. And so this wave of depolarization spreads across the muscle cell on both sides away from the motor end plate. As we depolarize, as the sodium gate hits -50, it opens up, brings in more sodium, and that, it will depolarize up to +30, and that will cause the adjacent gate next to it to depolarize, and the one next to it, and the one next to it. And then we have this repolarization event that occurs right afterwards. It's like, you ever see any, if you had a row of dominoes standing up and you knocked over the first domino, what's going to happen? They're all going to fall down in sequence. You've probably seen that somewhere, so they all fall down one after another. If you didn't touch them, nothing, they would just continue to do that. Imagine if the dominoes could fall, and after they fell, they could stand back up again, ready to do it again. So this sequence of depolarization and repolarization spreads across the muscle cell down to all parts of the muscle cell, down to every myofibril, down to every sarcomere. It comes down to T-tubules. So every part of the muscle cell is exposed to the action potential, the depolarization wave that runs down across the membrane. So this is the action potential. That's all well and good, but what does that have to do with a contraction? Okay, so we have another phenomena called excitation contraction coupling. The process of sending the action potential across the cell membrane is tied directly to the contraction step. We still haven't gotten a contraction, but we're getting there. It's a good thing this is fast, because if you're like, okay, there goes the action potential down my biceps, I think I can move my arm now. This happens in milliseconds. Remember that a depolarization and repolarization takes about 7 milliseconds, 7 thousandths of a second. To us, it's instantaneous. So we've got our action potential going across into the muscle cell. It goes down into the T-tubules. The T-tubules are connected to the terminal cisterns on each end of every sarcomere. The action potential, actually, the depolarization actually causes the terminal cisterns to open up. The action potential brought down the T-tubule causes terminal cisterns to open up and flood the entire cell, all the sarcomeres, with calcium. So here we've got, let me bring up that camera here. Okay, here's our model of this is a cell. Up here is the neuromuscular junction right there. And here we're seeing a whole bunch of sarcomeres. Sarcomere goes from Z line to Z line. The Z line is that area right in the middle. This is the I band, the light band, the other light band. This is all actin. This is all actin. And in the middle is myosin. So sarcomere to sarcomere right here. Now, the T-tubules are these blue things. I don't know if they're blue or not, but they line up on the ends of the A bands. On each side of the A band, there's a T-tubule there and a T-tubule there and one there. Now, these tan structures here are the sarcoplasmic reticulum. And this bulge on each side of the T tubule, these bulges are the terminal cisterns. They're filled with calcium. The whole sarcoplasmic reticulum is filled with calcium. What will happen is that the action potential comes down the T tubules, because the T tubule is nothing more than an extension of the cell membrane. They'll come down here and open gates in these cisterns and allow calcium to flood out and cover every sarcomere in every myofibril in the muscle cell. So whatever is happening at as far as the action potential goes, it's going to affect the entire muscle cell. You know, the muscle cell is made up of thousands of myofibrils, and there's probably millions of sarcomeres in all those thousands of myofibrils, maybe billions, and they're all getting exposed to calcium at the same time. And the calcium is key for us now. So the calcium is going to allow us to have a contraction. So everything we've done to this point, the action potential coming down the nerve, the release of acetylcholine, the opening up of the sodium channels at the motor on the motor end plate, the localized depolarization, hitting the threshold, depolarizing the muscle cell itself, all that has gotten us to this point where we can release calcium. Because calcium is what we need to have a contraction. So we've done all that just to get to release the calcium from the sarcoplasmic reticulum. So.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potential that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T tubules make contact with the calcium-storing membranous network known as the sarcoplasmic reticulum, or SR. Where it abuts the T tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
Okay, so everything, as I said, everything we've done to this point is to get the release of calcium. Calcium is essential to the contraction. Now let's take a look and see what's going on here. Now remember that every depolarization event only takes about 7 milliseconds. So again, for us, it's instantaneous. I mean, we can barely comprehend the passage of time of a second, let alone 7/1000ths of a second. Okay, now, so what happens here with this calcium? Well, the calcium floods out into the sarcomere. Now, if you remember, the sarcomere has actin, the thin filament, on either side of each band of thick filament, the myosin. The myosin Proteins are large large structures made up of about 300 molecules. Each thick filament is made of about 300 molecules of myosin. Myosin has two heads on it that look like drivers of golf clubs. That's the best analogy I can make for it. And the actin is made up of three parts. It has the actin, the thin filament is the actin itself in a double, it's moving in like a double helix structure. And woven in through the actin is tropomyosin, and attached to the tropomyosin is something called troponin. Now, the whole idea to get a contraction is to allow the myosin heads to grab the actin and pull the actin on either side of the A band towards the middle of the sarcomere. The actins are attached to the Z lines, and we're ending up shortening, we're pulling on the Z lines, and that causes the sarcomere to shorten. And this is going to happen in every sarcomere, in every myofibril throughout the muscle cell. Problem: tropomyosin is conveniently located on the actin where it blocks the binding sites. There's a special binding site on every actin molecule where myosin can grab onto it, but the tropomyosin runs across that and blocks the myosin from attaching to it. As long as the tropomyosin's in place, the muscle is relaxed. However, this is where calcium comes into play. Calcium binds to troponin. Now, troponin is attached to the tropomyosin. And when calcium binds to the troponin rolls over and drags the tropomyosin with it. This exposes the binding sites on the actin. When the binding sites for the actin are revealed, then the myosin heads will automatically reach up and grab onto the actin and give it a good yank on either side of the, the A band, and pull the actins towards the middle of the A band, towards the middle of the sarcomere. And in doing so, it shortens the sarcomere. And as long as calcium is present, then those binding sites are going to be exposed. As soon as calcium goes away, the binding sites then get blocked. The tropomyosin rolls back over into place. As long as we're sending a signal to the cell, we're going to keep pumping, we're going to keep releasing calcium. The binding sites are going to stay open. The myosin heads will keep on grabbing onto it and pulling it towards the midline of the sarcomere. So we have 4 steps to this. We have what's known as crossbridge formation. That is where the myosin heads attach to the actin. These myosin heads have been charged up with ATP. They have energy. They are in what we call the cocked position and ready to fire. Once they attach to the actin, they will pivot forward and they will pull the actins over top of the myosins. This is why we call it the sliding filament model, because the thin filaments actually slide over top of the thick filaments. So that's the power stroke. They pull the actins over top of the myosins. Now the myosins are stuck there now. They're stuck attached to the actins. But ATP comes along and attaches to the myosin and when the myosin head when the ATP attaches to the myosin heads will let go, drop down. They will then recock and they're ready to go again. So it's a 4-step process. We form the cross bridge. We have the power stroke. ATP attaches to the myosin head and that allows it to let go of the actin. It'll drop down. And then the last step is to recock, ready to go again, to reach up and grab the actin. And as long as you've got ATP, and as long as there's calcium there to keep the binding sites exposed, the myosin heads are going to keep reaching up and yanking, keep reaching up and yanking. Oops.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the Crossbridge cycle. In the skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads and thick myofilaments form cross bridges with actin molecules and thin myofilaments. The formation of a cross bridge is initiated when calcium ions released from the sarcoplasmic reticulum bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1, cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2, the power stroke. ADP is released, and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3, cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4, reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
So as long as we have calcium, we're going to be able to expose the binding sites on the actin. We also need ATP. ATP is what allows the myosin head to have the power to give the actin a good yank towards the middle of the sarcomere on, from both sides, and then let go and recock. If we run out of ATP, the myosin heads will be stuck in the, they have pivoted and they have pulled the actin towards the middle of the sarcomere. And now they're stuck there. There's no ATP available. So we've got to have the ATP for it to let go and recock and do it again. It's the only way that we can contract. Now, if we run out of ATP, well, we can't, we're stuck. If the calcium levels drop, then the binding sites get blocked and no more contraction either. So it's a lot of things that have to go on here. So everything up until the point, the whole action potential and the depolarization event brings us to the point of releasing the calcium, because without the calcium, we can't expose the binding sites. The binding sites aren't exposed, we can't get a contraction. Now, the reason I spent so much time talking about action potentials and depolarization and repolarization is because when we move into the nerves, in the nervous system in the next chapter, they do it exactly the same way. They don't deal with calcium, but they generate their action potentials exactly the same way as the muscles do. So, yeah, let's go on to the next slide. So what's happening here, if you want to see it, just picture by picture. The blue line there is tropomyosin. It's on the bindings. It's lined up, woven through the actin. Now the actin are those pink and red balls there. It's a double helix of actin protein. The binding sites are those light areas on there. The tropomyosin is blocking the binding sites. These are the binding sites right here on top of every actin molecule. They're blocked by the tropomyosin. These blue band, these are proteins. I don't think they're blue, but they're blocking the binding sites. The myosin head can't get to the actin. The calcium will come in. If calcium is present, it will bind to the troponin right there. When calcium binds to troponin, it changes the shape of the troponin molecule, causing it to roll over, exposing the binding sites so the myosin can grab onto the actin. And once the myosin grabs onto the actin, it will be able to pivot towards the middle of the sarcomere, dragging the actin with it. Because remember, the actin moves, the actin slides across the myosins. The myosins, other than the myosin heads that are going to be moving, the myosin never changes, it never moves around. The actins will slide over top the myosins, and then when they relax, they'll slide back. Now, what you're actually seeing in this picture here, you can see the thick filaments here and the thin filaments there, and you can see the myosin heads attaching to the thick filament, to the thin filaments. There's a myosin thick filament there. There's the actin right below it. These are the myosin heads attaching or detaching from the actin. So the myosin head will grab onto the actin, give it a yank towards the midline. It will pivot towards the midline, dragging the actin with it, and it will remain stuck there until another ATP comes along. As soon as an ATP comes along and binds to the myosin head, it will let go, drop down, recock itself, ready to do it again. The ATP allows our myosin heads to detach and reset themselves. Now, about half the myosin heads are contracting at any time. The other half are getting ready to contract. So each myosin filament, each thick filament has about 300 myosin heads in there. And it has 600 myosin heads. Because they have two heads, there's about 300 myosin molecules in each thick filament. So at any given time, about 300 myosin heads are contracting and the other 300 are getting ready to contract. As long as we have calcium and as long as we have ATP, the myosin heads are going to reach up, yank the actin, drop down, reload, and do it again. And so we don't see a delay because half the heads are contracting, the other half are getting ready to contract. So to us, it's a seamless event. This is a nice slow animation of what's going on. There's the yank towards the midline. The myosin heads are still attached to the actin. ATP comes along, lets them detach. That's what's happening right here. They detach, they recock, raise up, grab onto the actin and pull towards the midline again. They can't let go until ATP comes along. So once ATP has come along and allowed them to let go and recock, then they will, as long as the binding sites are exposed, they will keep yanking on the thin filaments, and the sarcomere will get shorter and shorter and shorter. So, and this happens in every sarcomere, in every myofibril in the muscle cell. It only happens in muscle cells that are stimulated, and you may have a cell stimulated and nobody else around it. That cell is going to be stimulated, and maybe a cell 100 cells away will be stimulated. Yeah, in the muscle, because it's there is a sort of a randomness to the order of these muscle cells. Now, each cell, when stimulated, is going to contract to its fullest extent that it can. And that leads us to the strength of the contraction. Contraction strength. So what is the fullest extent of that muscle cell? How powerful can that muscle cell be? Well, three things affect that. How many cross bridges can we make in the myofibril? How many bindings between actin and myosin can occur? How many myofibrils do we have in the muscle cell? Now, we can't alter the number of muscle cells. We don't grow new muscle cells. We're stuck with what we had when we were born, but we can alter the number of myofibrils. Exercise and protein consumption allows us to increase the size of the cell. As we eat more, as we exercise those muscle cells and we take in more protein, we can make more myosin and actin. They're just protein that we can make. And the muscle cell will get larger, it will get stronger, it will have more actins and more myosins. So we can enlarge it, or we can do no exercise and eat poorly and have few myofibrils. We can have small myofibrils with, just few actins and myosins in there. It depends on, what our diet is and what kind of exercise we have. And then the third thing that affects the strength of the contraction is how many muscle cells do we actually bring to play? If you're going to shake hands with somebody and you're not engaging in some sort of one-upmanship, you're going to shake hands politely, if you will. Are you determined to crush their hand when you shake their hands, or are you going to do it normally, gently? What do you, are you a crusher?
Depends on who it is.
It depends. Cameron, are you a crusher? I didn't think so. But some people are crushers and some people are gentle. How do you know, how do you keep from crushing somebody's hand? Is it something that happens automatically, or do you think about it, I'm going to gently shake your hand? It's a voluntary action. How many muscle cells do we have to use to gently shake somebody's hand, or to drive a nail? Or if you're putting in a nail in the wall to hold a picture, and you have one of those little, hook and nail gizmos that you use to put a picture up, like what's on the back wall back there, are you going to use a little hammer and tap it in gently, or are you going to get out your 20-pound hammer and, wham, punch a hole in the wall? I got it, How many muscle cells are you going to use? We are going to use the number of cells it takes to get the job done. We generally don't overdo it. Sometimes we do, but generally we don't. You're driving down the road and the light turns yellow, and you're not going to be able to get through it. So you push on the brake pedal and you slow down and stop. Okay. Now, if you're driving on the road and the person in the car in front of you stops suddenly, you put your foot on the brake pedal. Are you going to put it on with the same strength you put on for the light turning yellow? And, or are you going to hit it as hard as you can? You're going to hit it as hard as you can. You used more muscle cells to do that. A little more intense urgency there. Intense urgency. I guess it works. Okay. So the strength of our contraction of our muscles is how many cross bridges can we make between the myofibril in the myofibril. That's a function of how big the myofibril is. The number of myofibrils, that's a function of protein and exercise. How many cells are we using? That depends on what we're trying to do. So the strength of our contraction depends on all three of these things. We can't do anything about the number of cells, but we can do a lot with how strong the cells are or how weak the cells are. So how do we get the whole muscle to contract? Well, we have to stimulate a lot of cells in the muscle. Now we know that every muscle is innervated by at least a motor nerve. Actually, there are several there are many motor nerves that are going to innervate a muscle. Each motor nerve can stimulate hundreds of muscle cells. The motor nerve can break up into numerous axons, which then will lead to the actual motor neurons, the individual cells, and each cell can have axon terminals. Generally, we like to say that we have one motor neuron to about 300 or so muscle cells. So one motor neuron can control about 300 muscle cells. Each muscle cell has one neuromuscular junction. Only one. It's only one place for the axon terminal of a motor neuron to connect to the muscle cell. So every muscle cell is individually controlled by an axon terminal. Now it may be, that axon terminal, this is an axon terminal, is controlled, maybe, you have one neuron controlling 300 axon terminals. So those cells being supplied by that neuron are all going to get the same signal at the same time. We call that the motor unit. The motor unit is the number of muscle cells controlled by one nerve. This is what we call the motor unit. A motor unit means that all the cells connected to that motor nerve are going to contract together at exactly the same time. They're going to be stimulated at exactly the same time. The action potential coming down the motor nerve branches out into the axon terminals, and every axon terminal releases acetylcholine at exactly the same time. And we get our depolarization in 300 muscle cells at exactly the same time. And we expose the binding sites at exactly the same time. And the myosins all yank onto the actins at exactly the same time, because the signal comes down simultaneously to all 300. That's a motor unit. Now, if we want really precise control, we keep the motor units very small, 3 or 4 cells, 3 or 4 muscle cells. The muscles around our eyeballs, the motor units are very small, 4 muscle cells per neuron, because we want to have really fine control over the movement of our eye. Same in our hands, our fingers, to have the fine control we need to manipulate our environment. We're going to have very few cells per motor unit. So the fewer the cells in the fewer the muscle cells in the motor unit, the more precise we can be. We have, as it says here, with the greater the fine control. So that's what the motor unit does for us. Now, the motor units are not formed from a whole cluster of cells together. It's not like a fascicle of cells is a motor unit. We spread the motor unit out across the muscle. So there's probably a pattern, but it appears to be random throughout the muscle. So if the motor unit fires, all those cells contract, but they are spread throughout, so we get a complete contraction of the muscles, of the entire muscle. Now the muscle, you're probably not going to, if one motor unit with 300 muscle cells contracts, you're probably not going to notice it because there's thousands and thousands and thousands of cells in the muscle. But they've all contracted. May not have any result, but they've all contracted because they're going to contract from all the way all over the muscle. So we don't have, one of the advantages of doing it that way is we don't get 300 cells on the bottom of the muscle contracting together, and we get an uneven distribution of the force. We don't want that. We want to have it be, equal distribution of the pool of the contraction of the muscle. So here's what we see. We see a neuron connected to an axon terminal at each motor end plate. And so we can have up to 300 axon terminals on a neuron. We see these in the large muscles for posture, for example, or maybe our leg muscles, where, we don't have to be that precise. We don't have to have fine control for standing upright. So our back muscles, same thing. We're not too worried about fine control. But if we're getting into our eyeballs or our hands, we're going to have fewer muscle cells connected to the motor unit. So now here's something, one of the advantages of having the muscle cells, the motor units, the cells of the motor unit scattered throughout the cell is it allows us to generate something called muscle tone. Now, I will tell you that maybe even on the final exam, there is a question about muscle tone. And one of the distractors, one of the wrong answers says that muscle tone is the pleasant sound our muscles make during exercise. Sounds reasonable. I've actually had people mark that as the correct answer too, even after I stand up here and say, that's not the right answer. Muscle tone is what happens when a motor unit fires and 300 cells contract and the muscle doesn't move, but all those cells have contracted because it takes more than 300 cells to get the whole muscle to contract. So the muscle doesn't move, but what it does is it creates a little bit of tension in the muscle. It also warms up the muscle, keeps the muscle warm. And then another motor unit fires and another 300 cells contract. They contract fully, but it's not enough to move the muscle. And then another motor unit contracts and sort of random throughout the muscle. So what it's doing is creating a little bit of tension in the muscle cell, in the whole muscle, making the muscle stay firm. It's holding the joints together. It's also keeping the muscle warm. Warmer muscles are more efficient than cold muscles. When we exercise, we generally have a warm-up phase anyway. So this is how our muscles try to stay warm on their own, even before we try and do any kind of warm-up. This tension that we rotate it around the muscle itself, it's like on the anterior surface, the posterior surface, the medial surface, the lateral surface, and it's just sort of random throughout here. We're not aware of it. The muscle is. This slight tension keeps the muscle in place. It holds the joints together and it keeps the muscle warm. It keeps the muscle ready to contract when we're going to have the whole muscle contract here. So this is called muscle tone. And you're going to hear the term tone again more in AP2 when we talk about nervous control of muscles, a smooth muscle. So, okay, now, okay, here's something else to consider. You know, we've all said, oh, I got a twitch in my eye, or, you know, we've all said we've experienced a muscle twitch. And that's not an incorrect statement, but a muscle twitch is something that is more theoretical than anything else. Because when we talk about a muscle twitch, we're talking about a single contraction of a muscle cell as a result of a single action potential. And generally, we don't do it that way. We generally don't have a single contraction of a cell, of one cell only, with only one action potential. We don't, our neuromuscular system doesn't operate that way. So the muscle twitch, though, because it is a simple contraction of the cell and relaxation, and then that's it. But the twitch does, in its theoretical state, allow us to see the various phases of the contraction process. And that's going to lead us on into something else here. Now, the first part of the muscle twitch is the latent period. It takes a few milliseconds to get the action potential down the T-tubules, causing the release of calcium from the sarcoplasmic reticulum, binding to the troponin, causing it to roll out of the way, allowing the myosin heads to reach up and grab the actin. That is the latent period. We get to excitation, contraction, coupling at that point. At the period of contraction, we have formed the cross bridges and we've pivoted towards the middle of the sarcomere. So we've got muscle tension. After that, we have relaxation. The calcium goes back into the sarcoplasmic reticulum and the muscle cell relaxes. So we have three phases of a muscle twitch. The latent period, where things have to happen to get the action potential across the muscle cell down to the T-tubules, opening the T-tubules, releasing the calcium, binding the troponin, and rolling the tropomyosin out of the way takes time. Forming the cross bridges takes a little time too. Relaxation takes a lot of time. So how much time are we talking about? Well, the latent period is about 4 or 5 milliseconds. Usually takes about that long to for our excitation contraction coupling to take place. Then we get our contraction phase. That can take 15, 20 milliseconds, maybe. And then we have a very long relaxation phase because it takes time to get all that calcium back into the sarcoplasmic reticulum. So this is a typical muscle twitch. Now, our muscle cells don't generally operate this way because we don't do, you know, a one and done. If we're going to generate an action potential, our muscle cells will generate an action potential followed by another action potential followed by another one. We just don't send one signal out to a cell and no other cells. But every cell will follow this same pattern: a latent period, a period of contraction, and a period of relaxation. And that period of relaxation is very long because it takes time to get that calcium back into the sarcoplasm. So now, okay, there's our twitch. Twitches are, when we have a twitch, it's not just one contraction, it's multiple contractions taking place. It depends on the location. Twitches in our eyes, the eye muscles twitch rapidly. Their excitation and contraction coupling phase is very, very brief. And the contraction phase is very, very brief. It's a very rapid contraction. Whereas if we're looking at a larger muscle, a muscle that's very, as I say, a fleshy muscle. I keep forgetting to take calf muscle out of it because that's not your calf, that's your behind, that's part of your hand. Anyway, they contract slower. They hold the twitch longer. A twitch in your muscle in your hand or in your calf, like when you're having, as we like to say, a muscle cramp in your calf, in your gastrocnemius. We're very aware of that because it hurts and it comes on slowly and doesn't go away easily. Whereas twitches in our eyes are annoying. We've all had them, right? Okay, good. So I'm not crazy. We've all had twitches and they seem to happen and they go away. They don't seem to hurt like something in our calf muscle does. We see there's different times involved. On the far left there, we have the lateral rectus muscle of the eyeball, and it is, it has the same latent period as everybody else does, but its excitement phase and its contraction phase and its relaxation phase are short. So it can get ready to do it again because it wants to be able to contract and move the eyeball around. The gastrocnemius also has the same latent period. It has a short contraction excitation phase, a very brief contraction phase, and even its relaxation phase is not very long. It's done by 80 milliseconds. Again, that's 80 thousandths of a second. We still can't comprehend that. And the soleus muscle underneath the gastrocnemius is slow all the way around, 200 milliseconds before it gets back to its relaxed stage. So it depends on where the muscle is as to how fast it's going to be able to contract. But again, a twitch is an abstract concept. We don't deal with a twitch, but the phases in every type of muscle contraction, whatever muscle it is, they're all going to be the same. They're going to have a latent phase it's always at 5 milliseconds, because it still takes time to get to the excitation contraction coupling phase. That can happen rapidly. The more myosin heads you've got, the more actin binding sites that you can bind to, the more calcium that you've got will determine very quickly how rapidly your contraction is going to be. Your muscle will contract more rapidly and powerfully if you've got more binding sites. And so it says here that the muscle twitch is only seen in the lab or someone that has a neuromuscular problem. If you go on into healthcare and take more advanced courses, you might have might in a lab actually use muscles from animals and stimulate them to contract, and you will get a twitch. When I was an undergraduate, we did lots of gastrocnemius muscles from frogs. They work very well, and you can get them to twitch, and give you a twitch. So, but that is, it's okay. I didn't like frogs. No, actually, I do like frogs, but the point is that this is an abstract phenomenon. We don't normally see a single twitch in our bodies. One thing, but what we do see is not a twitch, but what we call a graded muscle response. This is the control that we have over the movement of our muscles. We control our muscles. We have different demands. The example of stepping on the brake pedal for different reasons, or shaking hands. You know, how do you shake hands with somebody? Do you crush their fingers or do you do it very gently? You know, are you engaging in a power struggle with someone, or are you just being polite? So this graded muscle response tells us how strong our contraction should be. We're not going to, you know, we're going to open the door with one set of muscles, and we're going to use our keyboard with a different level of strength. You're not going to be banging on your keyboard or pressing your touch screen with the same strength that you would drive a nail, unless you're really upset with your computer and maybe you want a new computer. I don't know. So anyway, we can control the strength of the contraction two ways. We can change the frequency of the stimulation, or we can change the strength of the stimulation. When we get into the nervous system, we're going to see that intensity of a stimulus isn't based on a change in the action potential. Intensity of a stimulus is based on the frequency of the action potentials coming from the sensory receptor. That's frequency of stimulation. If you bang your thumb with a hammer and it hurts, you're getting action potentials that are exactly the same size all the way. You know, every one of them is identical. Every action potential in our body is identical to any other action potential, regardless of where it comes from. But we measure intensity by how rapidly we're receiving the action potentials. It works the same way with our muscle cells. The faster we stimulate the muscle, the stronger the contraction is going to be. We can change the frequency of the stimulation. We can change the strength of the stimulation. So these graded responsive responsive muscle. I like that. So responsive. Okay, you can contract your muscle with any strength that you've got. You can be gentle. You can max out the strength depending on what force you can apply. We vary that all the time. Every second of the day, we are varying our responses. We alter how often we stimulate the muscle cells. We aren't even thinking about it. What we're saying is, I want to pick up my phone and put it back down. Well, we didn't stop to think, okay, I need muscle cells 500 through 3000 on the brachioradialis to pick that up and put it over here. We don't have to do that. We already know what we're going to do. We think, we generate the action potentials in our brain and we go ahead and do it. Now we have a series of smooth contractions that we didn't stop to think about. It's under voluntary control, but we don't have to control every muscle cell to get it done. Now, to achieve this smooth effort, these smooth contractions, we can vary the frequency of the stimulus, meaning we can stimulate and then stimulate somewhere else and stimulate somewhere else, or we can recruit. We can bring in more muscle cells to do the job, or we can take away muscle cells to do the job. If you're picking up a 5-pound bag of potatoes, you're going to use a series of muscle cells to do that. If you're picking up a 50-pound bag of potatoes, you're going to have to bring in a whole lot more muscle cells to do that. That's recruitment. Now, if we're going to do graded muscle response by changing the frequency of the stimulus, if we increase the frequency of the stimulus, we're going to see a phenomena here called wave summation or temporal summation. Temporal means time. It's summation over time. If we hit the muscle cell with the stimulus, and then hit it with a second stimulus, it will have a greater contraction strength. If we don't let the muscle cell relax between the first and the second stimulus, we'll get a greater contraction for a variety of reasons. We still have calcium surrounding the thin filaments. The muscle is warmed up. We've also exposed more binding sites, because if we have additional calcium present, it will bind to more troponins and roll over the tropomyosin. So we'll have additional binding sites present to have more myosin heads attach onto it. Not every myosin head is going to be able to attach to every actin in a muscle cell. We have our nerve impulse come down, our action potential comes down, we release calcium, it floods in there, and we bind the troponin, and the troponin rolls out of the way, and the tropomyosin moves, and we have binding sites exposed. But did we expose them all? Maybe not. You know, how much calcium did we have out there? Well, if we add more calcium, we expose more binding sites, so we can have more myosin heads grabbing onto the actin and giving us a greater second contraction. And if we keep adding a stimulus without any rest period, we continue to release more and more calcium, and the contraction can get stronger and stronger and stronger because we are exposing more and more binding sites. And it looks something, consider this. Here is a muscle twitch. We've seen this already. There, you know, 3 separate muscle twitches, takes about 200 milliseconds or thereabouts for each twitch to take place. We have the latent period, we have the contraction, and we have the relaxation phase. On the first one, we hit the muscle cell at time 0, and we're relaxed somewhere about 200. So at 200, we give the muscle cell a second stimulus, and it contracts again. Notice that the amount of tension formed on the second stimulus is just like the tension on the first one. There's no difference, essentially, from this level of tension here and this level of tension here. If we let this muscle cell then relax down for another 200 milliseconds, and we hit it again with a third stimulus, we get the same result. We get tension that is equal to the tension from the first 2 stimuli. We contract, we relax. We contract, we relax. But what if we change the rules? What if we hit the muscle cell with a stimulus? There's our contraction. It comes down to 0 again as far as tension. We hit it with a second stimulus. Only this time, we don't wait for it to relax completely. This time, we hit it with a stimulus before it's relaxed. It still has plenty of calcium in around the actins. Now, with the second stimulus, we dump more calcium out into the sarcolemma, into the sarcoplasm. More calcium is present to bind to the troponin. More tropomyosin moves out of the way. More binding sites are exposed. More myosin heads can grab onto the actin. So our contraction is stronger in the cell because we have more binding taking place. So we get a second contraction that's stronger. And if we let that relax, it takes, you know, more time to relax too. But what if we didn't let it relax? What if we hit it with another contraction here, like we're doing over here? So here we hit it with 1. We don't let it relax. We hit it again. We don't let it relax. We hit it again. And we keep doing this. This is wave summation or temporal summation. We are adding up strengths of the contractions, making them more powerful each time because we are releasing more and more calcium into the sarcoplasm. We are getting the muscle cell warmer than it was before. we're exposing more binding sites, and the myosins can grab onto more and more actins and create a greater strength of greater force of contraction because there are more places to attach to. And so we can continue doing this until we achieve a maximum strength of contraction. We generate the maximum tension where you can't generate any more strength. We call this tetanus, not like the disease, but this is tetanus. The muscle is reaching its maximum level. Now, if we can increase the stimulus even more, we will have complete tetanus. When we have a relaxation, when we do have a relaxation dip, we call this incomplete tetanus. If we increase the stimulus, then we get what's known as complete tetanus. The muscle has maxed out. It can no longer generate any more tension. It is at its fullest strength, its maximum strength. And you're lifting up 1,000 pounds over your head. You're holding it up over your head, and you're using every bit of muscle power that you've got to hold it over your head. And then the muscle will become fatigued and you'll drop it. That's what's happening at the end, where we hit fatigue and the muscle relaxes rapidly. We increase the rate of stimulus to the point there is no relaxation phase. We generate maximum strength that our muscle is capable of doing. And that's a function of how big are the cells, how many actins and myosins that they have, how much ATP is present, and how much calcium's there. Now, what we've done is we've loaded up the sarcoplasm with a lot of calcium. Every time we stimulate the muscle cell, we're causing the release of more calcium, which is binding to more troponins, causing it to roll over out of the way and bind the tropomyosin out of the way, exposing even more binding sites. If we've got a boatload of actins and myosins, we're going to expose as many as we can. And the more we expose, the stronger our our force is going to be. Because we've got lots of actins and lots of myosins, we can keep binding and binding and binding. So all because we're exposing more binding sites as we go along. This is complete tetanus. This occurs when we are, you're picking up something extraordinarily heavy. You're trying to achieve your goal of being an Olympic weightlifter, for example, and you're practically straining to lift something, and you're bringing all your forces to bear. Now, don't get tetany confused with treppy. Treppy is a German word for staircase. And lots of times people get these two mixed up. Treppy is where you do allow you generate tension in your muscle cells, and then you allow them to relax, and you do it again, and you do it again. What's happening is that this is what we see in warm-up exercises before some sort of athletic event, baseball, football, basketball. You have warm-up exercises. You have warm-up if you run track or cross country or swimming. You stimulate your muscle cells, let them rest, stimulate them again, let them rest, stimulate them again. It warms them up, increases the level of calcium in the sarcoplasm around here. You just don't generally, start running a marathon. You just walk, walk up to the starting line and say, okay, I'm going to run this marathon without any kind of warm up. Because your muscles it's going to be very painful for your muscles. You're, unlikely to finish it. You have to warm up here. And this is what treppy is. Treppy is the warm up phase that we do before we start exercising. We go to the gym and work out. We have some warm-up exercises we do just to get our muscles acclimated to the activity we're going to do. You don't go on a 50-mile bike ride without a little bit of warm-up. Or you don't go on a hike in the mountains without a little bit of warm-up. It would be very unpleasant. So this is called TREPI, because what we see, the staircase effect, we see the contraction strength getting greater and greater and greater. It's not tetany, because there's a relaxation phase in between each one of these. Now, so that was wave summation. We can increase the frequency of the stimulus to get a greater and greater contraction. The other way that we can control our muscle response is to bring in more and more muscle cells. Now, we start out small. We start out with the smallest motor units first. We keep adding motor units until we achieve our goal of moving what we're trying to move, or trying whatever, as we say, manipulate our environment. We start out small and keep going up and bringing in more cells until we can accomplish our goal. Because it certainly isn't productive to start out massive. Why kill a fly with a 20-pound sledgehammer? When a rolled up newspaper will do the same and won't leave a hole in your wall. Why stand on your brakes just coming to a stop sign when a general application would be enough? We always start with the smallest motor units and then work up, so that we don't, because if we use the largest motor units first, we have much less control. We have great control over the small motor units. The larger motor units are more for posture and position and stuff like that. Why do we want to bring those in if we don't need to? Okay. Now here's this is a good thing to finish with today too. Rigor mortis. What happens to a body after death? Our bodies will stiffen up. They will start to stiffen up 3 or 4 hours after death. It starts from the head and works down. After 12 hours after death, we are stiffened into the position that we died in, and that can be any shape, and will stay that way for about 2 days, 48 hours, 72 hours, maybe 3 days. So what happens here is, after death, the muscle cells can survive for several hours. Muscle cells have lots of stored oxygen present in the myoglobin. That's what makes our muscles red. They've got lots of glucose stored in the glycogen. So cells can generate a lot of ATP for a while. And so what will happen is, as we run out of ATP, as we run out of oxygen, and we run out of ATP, the myosin heads, lose their ability to let go of the actins. Now, the more calcium, and this is that phenomenon about calcium, the more calcium that we release into the dead cells, as the cells are dying, we release more calcium. So let me back up before I get too excited about this. Normal healthy cell contracts when calcium gets released. I just went through this whole thing about tetany, about how the greater the frequency of stimulus, the more calcium is available and the more binding we get. Well, consider this at rigor mortis. The cell isn't getting any more calcium. The cell is gradually dying. Muscle cells can stay alive for up to 12 hours before they die, the individual cells. As they start dying, the sarcoplasmic reticulum starts releasing more and more calcium. That opens up more binding sites on the actins. the myosin heads will reach up and grab onto them and yank the actins. As long as we've got oxygen and glycogen, we will make ATP and the myosin heads will let go. But as we run out of oxygen, and as we run out of glycogen, they'll contract and they won't let go. And as the cell starts to break down, more and more calcium gets released, more binding sites are found, more myosin heads attach, and the muscles start to constrict tighter and tighter and tighter. And at the end of 12 hours, we are locked in position. There is no detachment of the myosin heads because we're out of ATP. And they'll stay that way for about 3 days. And then they will, the proteins will break down they'll degrade thanks to bacteria. So whatever position the body has died in, if they're not moved, well, they're going to go into rigor, whatever position they're in at the end of 12 hours. So what we see is the calcium gets released as the sarcoplasmic reticulum breaks down. We get more and more binding, and eventually we run out of ATP and glucose. We run out of glucose and oxygen, so we don't make any more ATP. The myosin heads stay attached, and they don't break down. So, like this individual here, and the female on the left, whatever position, for whatever reason, how she died, I don't know. The individual on the right looks like he may have been engaged in some sort of recreational pharmaceutical, and he was found dead in position with his hand in the air. We also see the one on the left had his hand on a table beside his bed and died that way. The woman on the right was probably a suicide. A lot of times individuals that will kill themselves with a shotgun in the mouth, which is a common way to go, will be found holding the shotgun in this position. And that's the position they're found in. And that's, she's dead. Her arms are sticking up in the air. Rigor is so powerful that you could take a corpse on a gurney, on the cart, and they could be in full rigor, and you could swivel the person off, so that their legs extend off the end of the gurney, and their legs would stick straight out, and there would be no sagging or anything, because the muscles are stiff. And so this is a phenomena. This is a very good way to determine the time of death too, because full rigor takes at least 12 hours to form, and takes 2 or 3 days to break down. So, and that is a great place to stop. So, anybody have any questions? So, okay, let me get out of this. And lab this afternoon, we're going to look at the second half of the general census. We'll talk about the proprioceptors, and we'll talk about reflexes and the different types of reflexes. So, and no one has any questions or comments, I'll get us out of here.